Good morning, scientists, and welcome back to our channel. It's always beautiful to have you here as we continue to discuss all our different concepts in science. We have been discussing about PCR, polymerase chain reaction, and we'll continue this our discourse by looking at what exactly is the effectiveness of this technique. Like we said in an earlier video, which you can check on any of our platforms, we explained that the PCR is a laboratory technique where we try to amplify, make many copies of a section along the length of a DNA. And there are many reasons for doing this. Now, in this video, we'll go into about 7 of the major reasons why we as life scientists want to run a PCR polymerase chain reaction, and what is the usefulness, what exactly are we doing with this procedure. Now, number one on the list is disease diagnosis. We use PCR to diagnose many different kinds of diseases. Now, when it comes to identifying pathogens, it's important to note that molecular techniques are the gold standard for identifying different kinds of pathogens, and PCR is at the top of the list of the methods that can be used to identify different kinds of pathogens. So when you have pathogens, which could be viruses or bacteria, now we have, depending on whether it's a virus, whether it's a fungi, whether it's a bacteria, we have sections. Like, for an example, if it's a bacteria, we usually use the 16S rRNA gene. If it's a fungi, we use the ITS region. So what we are saying is that there are sections along the DNA of whether it's a bacteria or a fungi or a virus. These sections are the ones that we amplify and then run them against already established database. So you have a sequence on a bacteria or on a fungi or on a virus. Now you amplify that sequence, make many copies of it, and then you now have the individual nucleotides. You know what the individual nucleotides are, and then you take this information and run it against a database, the NCBI database, and that way you're able to identify that organism. So we use PCR to do a lot of identification in the field of medicine for medical diagnosis. So COVID, different types of illnesses, we're able to identify them using PCR method. Secondly, we use it in genetic and genomic research. So we can have different genes that we want to identify. We want to see which gene is doing what particular function. So in these kind of instances, also if we want to manipulate genes, and when we say manipulate genes, we mean we want to change around the structure, the nucleotide sequences in genes. When we want to do this sort of analysis and this sort of work, we need PCR. We use it to identify our gene of interest, to identify the individual nucleotides that make up that sequence of that gene. So we use PCR a lot in genetic and genomic research. We can also use it for mutation identification in genetic research. So if there is a mutation, and you know what a mutation, that is where you have a situation where there is a change in the nucleotide sequence, and this change can lead to many different conditions. It could be a silent mutation such that this change doesn't necessarily bring about any change in the phenotypic characteristic of the organism, yet it could be a change that leads to different other conditions. It could lead to illness, it could lead to change of genetic traits. So we use PCR to study mutation under genetic and genomic studies. This is really important for us to know. Also, in the field of microbiology, just as we do in medical research, in microbiology, we also use PCR to identify different kinds of pathogens. Not only pathogens, we use it to identify different kinds of organisms. Organisms that have importance in environmental research, that have importance in agricultural research, that have importance in public health research. All of this we identify in microbiology using PCR and using our knowledge of PCR. This is really important. PCR is also important in forensic science. In the event that a crime has occurred, somebody has been raped, somebody has been killed, whenever a crime occurs and you have mixing of body fluids, to be able to identify the perpetrator, to identify the rapist, to identify the culprit, we use PCR. We use it to identify the genes of different individuals that are involved. We have genetic gene fingerprint, DNA fingerprinting, and different other techniques that we use. And PCR is nested in the middle of all these techniques, and it helps us to be able to identify perpetrator. So it's used a lot in forensic science to solve crimes of different types. PCR has importance there. PCR is also used in agricultural research. Different kinds of when you want to have modification of different kinds of agricultural products, different fruits, different berries, fruits, foods, when there is need to first to identify the organisms, the food organism, the plant organism, sorry, and then if there is need for mutation, if there is need for change of to in order to get desirable characteristics, PCR sits in the middle of all of this because it is used to identify genes of interest, making it important in agricultural research, in medical and clinical, also in environmental and ecological studies. Once organisms are involved and there's a need to identify those organisms, PCR along with gene sequencing can be used as the gold standard for identification of different kinds of organisms. This we spent some time looking at the usefulness of PCR. Why is it that we in the life sciences want to use PCR? We will do a video, a follow-up video where we'll talk about the different types of PCR. As of today, there are about 18 different types of PCR. So we'll break the video down into three or so where we'll talk about the different types of PCR. Scientists, don't get tired of this journey. Remember to like, subscribe to our channel and our content, and follow us on this beautiful science journey. You have a wonderful day.